Today, Jonathan and I are kind of going to both talk about different aspects of things we've been thinking about that may be able to be potential uses for benign urological defects, specifically the urothelial defects. And the idea is that perhaps there are urothelial defects that are associated with some of the benign diseases that progress, but may not be the primary cause of the disease, but may also contribute pain and other aspects to the disease as if they progress. So it may be possible to treat these defects in that context. Okay, next slide. How do I get to the next slide? Let's see. Okay, just a very short introduction. If you look down here, it's a schematic. This is the urothelium, the epithelium covering the bladder. Specifically, it goes from the renal pelvis to the bladder neck. It's a barrier. It prevents water and fluid exchange. It prevents infection. And these are very, very slow dividing cells, so that's something to keep in mind when we talk about regeneration and injury. There's 4 known cell types in the mouse and human right now. The superficial cells, which are the big polyploid cells that make the substance that provides a barrier, the uroplakin crystals, and intermediate cells, which you can see down here, which can be binucleated, and they will give rise to superficial cells during homeostasis. 2 populations of basal cells, the K14 expressing, which are very rare, 1 out of 20 in mouse, and the K5 basal cells, which make up about 80%. And you can see here, I just put this so you can see an idea of how big the superficial cells are compared to the other cells. And here's the intermediate cell progenitor. So quickly, what happens? How do we deal with injury and homeostasis? What are the progenitors? So during homeostasis, intermediate cells, as I said, can undergo failed cytokinesis. They can produce binucleated superficial cells. These have a very long half-life, can be 40 weeks, according to literature. They're repeated injury, for example, UTI, CPP, injury that actually denudes the urothelium, or even UTI just acute. Basal cells of K14 basal cells can be progenitors. They're probably the progenitors that do the de novo repair, and they can produce K5 basal cells, intermediate cells, and intermediate cells make superficial cells. And so these can divide and renew in after repeated injury. But there is another program that we've identified, which we call basal cell activation, that occurs in the skin and the airways, which is a response to injury, wound healing. And in this situation, the basal cell progenitor, instead of making urothelial cells, begins making squamous cells, and they express markers like keratin 6A that are never expressed in the healthy urothelium. So this can be a transient effect, or it can be a chronic effect, which can lead to other problems, and this may occur in aging, diabetes, LUTs, and chronic injury. So that's one of the things we want to look at, and we want to understand whether it's possible to fix some of these defects with reagents. So we've been working on PPAR gamma. It's a nuclear receptor superfamily member. These are all transcription factors that are activated by ligands. Natural ligands or fatty acids, for example. Synthetic ligands for PPAR gamma include rosiglitazone and pioglitazone, and some of these have been used to treat diabetes, but they do have side effects. And it's been shown that PPAR agonists can basically drive basal K14 cells in vitro to start to express markers of mature superficial cells. So they have a potent role in potentially in the urothelium, and we've shown that in vivo in mice, PPAR gamma loss of function actually leads to squamous differentiation. You can see here PPAR mutants, and PPAR gain of function. For example, if you express an activated form of PPAR in K14 basal cells, will convert the basal cells almost directly into superficial cells. And so you see the control superficial cells on the top, basal cells on the bottom. Here you see these superficial cells forming in the basal layer after expressing this K5 CRE-ER BP16 PPAR gamma, which is an activated PPAR gamma. So what we think happens is, in this case, the basal cells progenitors are making squamous cells. In this case, they're converting directly into superficial cells. PPAR gamma is also very important in cancer, which has two subtypes, kind of similar to what's in the endogenous urothelium. The basal squamous type of cancer, which has got expressed basal markers, also squamous markers, and the luminal cell type, which express markers of intermediate and superficial cells. So PPAR is downregulated in the basal subtype and amplified in the luminal subtype, which has led people to suggest that it might be important as a driver or suppressor of cancer. So can PPAR agonists be used to repair the damaged urothelium? Well, if you just focus on the left panel, the right is a control, we've actually shown that basal tumors, which regenerate in mice, can be basically annihilated by rosiglitazone, which is a PPAR gamma synthetic agonist. So if you look, this is an ultrasound of the same bladder, the same mouse, before and after rosiglitazone treatment. And here you see the ultrasound shows formation of a tumor here, which is in yellow, the tumor. And after this agonist, 1 month, these tumors are basically gone. And we actually find them gone after about 4 days. So we see that the tumor cells undergo apoptosis very shortly after PPAR gamma agonist treatment. And so here is just another slide showing you the regeneration of the epithelium, which is a separate entity, we think. So not only do we get rid of the tumor in the PPAR gamma agonist treated samples, but we also restore normal urothelial differentiation. This is very important because this is a problem if you have an inflamed situation. How do you restore normal differentiation? Well, PPAR acts on the immune system. It suppresses NF-kappa B signaling, and it also promotes urothelial differentiation. So those are two very important functions that could be used to treat benign disease, and that's our goal here. And we've also done ATAC and RNA-seq on shortly treated urothelium, and we see that a number of markers are upregulated, including markers of retinoic acid signaling, and as well as markers of the luminal differentiation program. And other things that are down-regulated include NF-kappa B signaling, chemokine signaling, as I just mentioned. So the idea is to determine whether these reagents are useful in a benign urological context. So in summary, what we see cyclin D1 down, P21 up. So this is probably part of the mechanism that kills the squamous, the basal subtype tumors. But we also see RA signaling downstream, which may be important in the luminal differentiation program, or alternately for suppressing the basal program. So as I said, what we want to do is try to repair urothelial abnormalities associated with benign disease. So we need models, and that's one of the things that I'm hoping that people will get back to me and tell me about your models, and we'll see. I'm not so sure about interstitial cystitis because it looks like it's very different, but perhaps diabetes, LUTS, overactive bladder might be good models. So now we're going to switch to Jonathan Barrage. He's going to do a talk about iron chelation as a potential mechanism. So here we go, Jonathan.
Okay, so I work with Kathy Mendelson, and Kathy's going to point for me, I think. And what one sees is that in the upper left-hand panel, Kathy's work, which demonstrates that NF-kappa B is activated within 2 hours of a UTI, by 6 hours the growth of bacteria, by 12 to 18 hours the shedding of cells, and 24 to 48 hours the repair of it with proliferation of cells. So I'm particularly interested in the first couple of hours of the response of the urothelium. You can see in the bottom left-hand side, you could see that at the 8-hour point, we see urothelial abnormalities, blebbing, and by the next point in time, the 12-hour point, that the cells actually fall off and are denuded. So we needed a way to find the genes that are involved very, very early on within the first couple of hours after a UTI. So we invented a new mouse that previous slide that I'm happy to distribute, that allows us to pull out RNA, nascent RNA, from any cell of our choice at any moment of time without any dissection of tissue or dissection of organ. And so we can pull out the nascent RNA expressed in the urothelium after a UTI, in this case 6 hours. The red genes are iron-related genes, and the yellow genes are heme-related genes. They're very prominent after a UTI. Next slide, please. So the two genes that we're most interested in, as shown at the top, is the NGAL gene, which is expressed at 4 hours after UTI. If you go over to the right side to the top panel, you can see that the NGAL knockout UTIs are very prolonged, even to 7 days, whereas in the wild type mouse, they're cleared in 4 days. The bottom panels show hemox. That's also highly expressed at 4 hours after a UTI. And you can see in the next panel right next to it, adjacent to it on the bottom, you can see carbon monoxide being released from the bladder. Go across to towards the right side. We invented a probe for carbon monoxide. It's the red signal at the bottom in the middle panel there. Over next slide, next over. And if you look at the hemox knockout, you can see that the cells are beginning to shed at steady state. And when you add a UTI all the way at the bottom on the right, you can find that the cells are rapidly shed. So both NGAL and hemox are necessary proteins in the early responses of the urothelium. Next slide, please. So this gave us the idea that we can control different forms of iron in the bladder, we can consequently reduce UTI. So you see an iron deficiency, the amount of bacteria that are present are quite reduced. In the bottom red bars, you can see that if we add NGAL in vitro, we can iron starve the bacteria. Those are all iron signals from the bacteria that are upregulated in the presence of iron starvation due to NGAL. And on the top right-hand panel, all the way over to the right, you see the NGAL crystallography that we did. When we resurface the blue amino acids to the red amino acids shown at the bottom of the crystallographic structure, we can actually give a subcutaneous injection of NGAL. It goes right into the urine, bypassing the reabsorptive mechanisms in the kidney. You could see the purple NGAL coming into the urine within a few minutes of the injection. And this is a way to deliver an iron chelator into the urine, very specific to the kidney. We could also do this with the heme pathway shown on the bottom. That's HEBP1 that we've resurfaced as well and can go right into the urine. So looking at the early damage pathways, finding critical genes in UTI, and then playing with them biochemically, we potentially have some therapeutics in iron chelation. Thank you very much.